Hi children, in video 1 we will be watching the structure of the DNA and the packaging of DNA in the chapter 6 molecular basis of inheritance. So going into the history of DNA. DNA was first identified by Frederick Mesher and he named it nuclein because it was found in the nucleus. He identified as an acidic substance inside the nucleus and described or gave it the name nuclein. He just identified, but he could not isolate it because of technical limitations at that time. So for a long time the elucidation of DNA was elusive, not disclosed, until 1953 when two different scientists isolated the DNA using X-ray diffraction data. The two scientists are James Watson and Francis Crick. Using X-ray diffraction data, they isolated the DNA and proposed the double helix structure. This double helix structure is now widely accepted model for the structure of DNA. There is a proposition put forward by Erwin Chargaff which contributed to elucidating the structure of DNA put forward by Watson and Crick. That is, the contribution of Erwin Chargaff also helped to describe the double helix put forward by Watson and Crick, and the contribution is the base pair rule. It can be represented in the form of a formula A by T is equal to G by C is equal to 1. It can be represented in this form, the base pair rule, or it can be told in a sentence or words. The ratio between adenine and thymine and guanine and cytosine is equal to 1 and is always a constant. So this base pair rule put forward by Erwin Chargaff helped Watson and Crick to make a complete description of the structure of DNA, and which is now called the double helix structure. So this contribution was very, very important and was a hallmark in the field of genetics. Clear? So once again, into the history of DNA, the person who first identified DNA but could not isolate it was Frederick Mesher. He named it nuclein because it was found in the nucleus. Then we have two scientists, Watson and Crick. They only proposed the double helix structure which is now widely accepted. And Erwin Chargaff's contribution to the field of genetics is the base pair rule. So that's all about the history of DNA. Now we'll go to the structure of DNA. Starting from the building block of DNA, we can construct the structure. The building blocks are the bases. The bases are adenine, thymine, guanine and cytosine. The bases that constitute the DNA are adenine, thymine, guanine and cytosine. And remember, adenine always pairs with thymine and vice versa. Thymine pairs with adenine by a double hydrogen bond. Similarly, guanine pairs with cytosine with a triple hydrogen bond or vice versa. And remember some more terms to this. A and G are called purines. T and C are called pyrimidines. This is very important to crack problems. If they ask you to find the percentage of purines and pyrimids, you must know of these which are the purines. The purines are A and G, and which are the pyrimidines T and C. So these terms are important to be learned for problems. I put all the 4 bases to build the DNA, to construct the DNA. Now the bases are connected to sugar. See, I am connecting all the bases with sugar, and the sugar in DNA is the deoxyribose sugar. Now this component is called nucleoside. So the next term we are learning is nucleoside. Nucleoside means sugar plus base. So what does the nucleoside comprise? Sugar and base. So this is one nucleoside, this is the second nucleoside, third nucleoside, fourth nucleoside. So in this DNA what I have drawn is, we are having how many nucleosides? 4 nucleosides, and accordingly they get their name. The name where adenine is a component of the nucleoside, it is known as adenosine. So adenine becomes adenosine. Where guanine becomes a part, it will be called guanosine. When thymine becomes a part of the nucleoside, it becomes thymidine. And when cytosine becomes part of the nucleoside, it becomes cytidine. It is not complete because it belongs to DNA. It is called deoxy adenosine, deoxy guanosine, deoxy thymidine, and deoxy cytidine. Okay, so these are the different nucleosides. So once again, what is nucleosides? The component of the nucleoside is base plus sugar, or base connected with sugar forms a nucleoside. So there are how many nucleosides? There will be 4 nucleosides. Why 4 nucleosides? Because there are 4 bases. When adenine becomes a part of the nucleoside, it is called deoxyadenosine. Look at the ending sin. When guanine becomes a part of the nucleoside, becomes deoxyguanosine. When thymine becomes a part of the nucleoside, it becomes deoxythymidine, and when cytosine becomes a part of the nucleoside, it becomes deoxycytidine. So these are the different nucleosides. Be familiar with the names. And because it belongs to DNA, we have to write deoxy. But if it's RNA, just remove the deoxy. Cut the deoxy, then it becomes part of the RNA. So if suppose these are the nucleosides of RNA, we will be learning about RNA shortly. What and all we know about RNA and DNA? Basic differences: DNA is double stranded, RNA is single stranded. Another difference we have learned is DNA has the bases adenine, thymine, guanine and cytosine, whereas RNA has the bases adenine, uracil, guanine and cytosine. So that's another difference. So here, if it's the nucleoside of RNA, it is adenosine, guanosine, but it's not thymidine. There's no thymidine. It will be called uridine and cytidine without deoxy. I hope you have understood the nucleosides of DNA and RNA. When you learn it, you have to learn for RNA as well as DNA. For DNA, we have to prefix deoxy. For RNA, just remove the deoxy and give the names accordingly. Adenosine when adenine becomes a part, guanosine when guanine becomes a part, then uracil when uracil becomes a part of the nucleoside, it's called uridine, and when cytosine becomes a part, it is known as cytidine. This is the case of RNA. But when DNA, when it becomes a part of DNA, we have to add the word deoxy, deoxy, and here for thymidine it's deoxy, and here deoxy when it becomes part of the DNA. I hope you have understood nucleoside. So once again, what is a nucleoside? Nucleoside consists of 2 components. They are the base and sugar. And when that nucleoside comprises adenine and the sugar, it's called deoxyadenosine. When it comprises guanine and the sugar, it is called deoxyguanosine. When it comprises thymine and the sugar, it is known as deoxythymidine. When it comprises cytosine and the sugar, it is known as deoxycytidine. It is in the case of DNA. But if it's in the case of RNA, you remove the prefix deoxy and just give the ending adenosine, guanosine. Then there's no thymidine. Instead of thymidine, it is uridine. And last one, same, remove the deoxy from cytidine. So you get it, the nucleotides for RNA. So coming back to DNA itself. So we are not going back to RNA. We are not giving a comparison with RNA, but here we were forced because we had to name the nucleosides. That's why I brought in the naming of the nucleosides of RNA. So, I am removing RNA from this template. Now, again we are going to build up the DNA, it is not complete. Now, this sugar is connected to a phosphate group. Now, this unit becomes nucleotide. So, what is a nucleotide? It is starting from your phosphate group plus sugar and base forms a nucleotide. So, here I am going to draw all the phosphates. So, we have one nucleotide, this is the second nucleotide, third nucleotide and fourth nucleotide. See, nucleotides, a number of nucleotides. How many nucleotides are there here? 4 nucleotides. So, we see a polymer of nucleotides. Polymer of nucleotides is called polynucleotide. So, we get the next term polynucleotide. So, what is a polynucleotide? When there is repetition of nucleotides, it is a polymer of nucleotides or polynucleotides. So, we can see in this diagram. So, we drew on one side. Now, the other side also should be completed. So, this one, what is indicated, I have circled, is one polynucleotide. Now, I am going to complete the other polynucleotide. To complete, first I must build the nucleoside connected to sugar. This is connected to sugar. So, now nucleoside is built. Now, from nucleoside I am going to build a nucleotide, just add phosphate group, and the phosphate is common, phosphate group. So, we have now 2 polynucleotides. See, this is one polynucleotide chain, this is the second polynucleotide chain. So, the DNA has 2 polynucleotide chains. Now, look at the endings of these 2 chains. See, can you identify the 2 polynucleotide chains? You can see here it ends with sugar and here it ends with phosphate. I will give another color to stand out. Okay, here it ends with sugar and here it ends with phosphate. Here it ends with sugar and here it ends with phosphate. This is very wrong. If here it ends with sugar, here it will end with phosphate, and here it should end with phosphate, and here it should end with sugar. I hope you have understood what I meant. The ending should not be the same. For example, I have highlighted the end of this polynucleotide chain is sugar and the end of this polynucleotide chain is also sugar. Here also it is sugar and here also it is sugar. This is very wrong representation. If here it ends in sugar, here it should end in phosphate group. And here it cannot be 2 sugars parallel. Here it should be phosphate and here it will end in sugar. So, this type of arrangement or alignment of the 2 polynucleotide chain is called anti-parallel. Or if I show it symbolically, this is called parallel. See the arrowhead parallel to each other, or the arrowheads up and the tail down, but this representation is called anti-parallel. So, the arrangement of the 2 polynucleotide chain is anti-parallel. The meaning of that is one end if it is sugar, the other end it is phosphate, and for the other chain one end will be phosphate and the other end is sugar. Now, to avoid confusion, there is a better version of this description, that is one chain run in the 5 prime 3 prime direction while the other chain runs in the 3 prime 5 prime direction. This will be made clear only when you learn the chemical structure of the DNA. As of now, we did not learn the chemical structure, just the skeleton of the DNA with its components. Clear? So, this anti-parallel alignment, one chain is running the 5 prime 3 prime direction and the other is running the 3 prime 5 prime direction, will be made clear. Why we are using the word 5? Why we are using the numerical 5 and the numerical 3? So, where it is phosphate, it is 5 prime. It is indicated as 5 prime, and where it is sugar, it is 3 prime. So, here it is 5 prime and here it is 3 prime. So, as of now, you remember where it ends with sugar, it is a 3 prime end, and where it ends with phosphate, it is 5 prime end. So, one chain is running in the 3 prime 5 prime direction, whereas the other chain is running in the 5 prime 3 prime direction. See, this is running in the 3 prime 5 prime direction, whereas this is running in the 5 prime 3 prime direction. Clear? This it is not parallel, that is 2 starting with 3 prime 3 prime and ending with 5 prime 5 prime. No, when one is running starting with 3 prime and ending with 5 prime, the other will start with 5 prime and end with 3 prime. As of now, you remember the phosphate group means 5 prime end and the sugar component indicates the 3 prime end. When we study the chemical structure, this will be very clear. Now once again a recap of what I have taken. The skeleton of the DNA. The DNA has bases adenine, thymine, guanine and cytosine. Adenine pairs with thymine with double hydrogen bond and guanine combines with cytosine with the triple hydrogen bond. Next, the bases are connected to sugar. See, on either side they are connected to sugar. See, this base is connected to sugar and these bases also are connected to sugar units to form the nucleoside. Then the sugars are connected to phosphate group. Then this component is known as a nucleotide. So nucleotide has 3 components, namely phosphate, sugar and the base. Clear? Like that way a repetition of nucleotides are seen. See, 1, 2, 3, 4. So we have 4 nucleotides, which means there is a polymer of nucleotide. This is one polynucleotide chain. Similarly, on the other side also there are 4 polynucleotide chains. 1, 2, 3, 4. 4 polynucleotide chains. So there is also one polynucleotide. So there are totally 2 polynucleotide chains that forms the DNA. And also remember how it is running. It is running anti-parallelly. When one chain is running in the 3 prime 5 prime direction, the other chain is running in the 5 prime 3 prime direction. It means that if one starts with sugar, it ends with phosphate, and the other starts with phosphate and ends with sugar. So to understand that, why we are using 5 and 3, we have to study the molecular structure. So let us go to the molecular structure. What I am drawing is the deoxyribose sugar. It has 5 carbons, 1, 2, 3, 4 and this is the 5th carbon atom and this is oxygen. And you have to remember at 1, 3 and 5 hydroxyl group is connected. So I am going to draw the hydroxyl group at 1, 3 and 5. Okay, so at the first carbon compound there is a hydroxyl group, third carbon compound there is a hydroxyl group and the 5th carbon compound I have drawn the hydroxyl group. Then as chemistry students you will know how to complete this carbon, satisfy the valence. There are 4 valence electrons, so satisfy them. So here 1, 2, 3, so 4, 1 hydrogen. Then here 1, 2, 3, 4. Here 1, 2, 3 and 1 hydrogen. 1, 2, 3, 1 hydrogen. So 1, 2, 3 and 4. Okay, so this is the structure of a deoxyribose sugar. Clear? So remember a deoxyribose sugar has 5 carbons and hydroxyl group is connected to the first carbon atom, third carbon atom and 5th carbon atom. It's easy to remember, at the odd numbers the hydroxyl group is connected. So this is a deoxyribose sugar. Now this deoxyribose sugar is connected to both base and a phosphate group. No, then only forms a nucleotide. We know what is a nucleotide. We have the sugar should be connected to the base as well as to a phosphate group. So we are going to make out of this I am going to convert this into a nucleotide. So this hydroxyl group is going to be replaced by a base. I am writing base. And this hydroxyl group, the hydroxyl group at the 5th end is going to be replaced by phosphate group. Clear? So now what we got? A nucleotide. So let's verify whether it's a nucleotide. See, phosphate, this is the sugar and base. Phosphate, sugar and base. So from the deoxyribose sugar, I replaced the hydroxyl group from the first carbon compound and the fifth carbon compound. From the first carbon compound, I replace the hydroxyl group and replace it with or substitute it with a base, and the fifth from the fifth carbon which was connected to a hydroxyl group, I replaced and substituted with phosphate group. Okay, now I got a nucleotide. Now using this digital board, I am going to make many such nucleotides. I am making it ready. So now I am going to draw the nucleotides. At least 4 nucleotides should be drawn for the exam. One nucleotide comprising A, second nucleotide comprising T, third G and fourth C. Any order, but we have to have the 4 bases, minimum 4 bases. So I have drawn the nucleotides and I am going to join it together. We know how we got a nucleotide. The first hydroxyl group was replaced by the base here, and the fifth hydroxyl group, that is the hydroxyl group connected to the fifth carbon atom, was replaced by phosphate. Now we got a nucleotide. So nucleotide, I got the phosphate, sugar and the base. So that's why I prepared 4 of them. Clear? Now what I am doing is I am joining them together. So this is my first nucleotide. To the first nucleotide I bring my second nucleotide. Now where will I connect this? This will overlap. See, this phosphate group will overlap on this hydroxyl group, which means the hydroxyl group on the third carbon atom is also going to be replaced by the phosphate group of the second nucleotide. Then this is my third nucleotide that is coming now. This will be overlapping the hydroxyl group of the third carbon atom. And my fourth nucleotide is coming. This fourth nucleotide also will be overlapping the hydroxyl group at the third carbon compound. So did you see the structure being formed? You have to draw and study the structure, then only you will get it. Okay, now I have my 4 and it's coming like stepwise down. And now I am replacing the base. Here it was written general base. Now I am replacing and giving the names. You can give it in any order, but I am going with A, base T, base G and base C. Clear? So I got my polynucleotide, which is having at least 4 nucleotides comprising A, T, G, C as bases. Now look at the endings. Now please focus at the endings. Look at this ending. At this ending I have phosphate group, and at this ending I have the hydroxyl group. So look at the free ends of this polynucleotide chain. At one free end I have the phosphate group, and the other free end I am having the hydroxyl group. Now you look, where is this phosphate group connected? This phosphate group is connected to the fifth carbon atom. See, this is the first carbon atom, second, third, fourth, and this is the fifth carbon atom. So the phosphate group is connected to the fifth carbon atom, and hence it gets the name 5' end. And look at the hydroxyl group. To which carbon atom it is connected? To the 3 carbon atom. That's why it's getting there ending as 3'. Now you understood why it is given 5' and 3'. The phosphate end is called 5' because the phosphate group is connected to the fifth carbon atom, and the other end which has the free hydroxyl group, it is not called the hydroxyl ending, it is connected to the 3 carbon component, therefore it is called the 3' end. So you understood, from the chemical structure you will understand why it is called 5', 3' end. This is very, very important, the chemical structure of DNA, and this DNA is running in the 5', 3' direction. The alignment is in the 5', 3' direction. Clear? I hope you have understood the structure of DNA, the chemical structure of DNA, which is very, very important for the board exam. And please indicate the 2 directions, 5', 3'. Where it's phosphate, it's called 5' because the phosphate is connected to the fifth carbon atom, and where it's hydroxyl group, it is called 3' because the hydroxyl group is connected to the third carbon atom. This is the chemical structure of DNA. Now getting back to the structure proposed by Watson and Crick, which is called the double helix structure. So, the salient features of the double helix proposed by Watson and Crick. The first point states that it comprises, that DNA comprises of 2 polynucleotide chains. So, DNA comprises 2 polynucleotide chains with the sugar phosphate units serving as the backbone. So, if I draw it very quickly, A, T, G, C, T, A, C, G. This should be always the pattern. See, I drew the polynucleotide chain with the 4 bases. Okay, A pairing with T, G pairing with C, T pairing with A, C pairing with G. Okay, so the first postulate or the first salient feature is the DNA comprises 2 polynucleotide chains with the sugar phosphate unit serving as the backbone. So, this is the backbone and this is the backbone. So, we have a tendency to say, taking our backbone in connection or relating to our backbone we think this is the backbone. No, actually the backbone is the sugar phosphate unit. So, as proposed by Watson and Crick, DNA comprises 2 polynucleotide chains with sugar and phosphate serving as the backbone and the bases directed inwards. See, the bases are directed inwards. Look at the bases, they are directed inwards. That is the first salient feature. So, once again the salient features proposed by Watson and Crick, the first one is DNA comprises 2 polynucleotide chains with sugar phosphate units. See this stretch and that stretch serving as the backbone and the bases directed inwards or protruding inwards. So, that is the first postulate or first salient feature. Second feature is the 2 polynucleotide chains are running anti-parallel. Very important, anti-parallel alignment. The meaning of this, you can write it symbolically. One is running the 5 prime 3 prime direction, whereas the other chain is running in the 3 prime 5 prime direction. So, here we will check whether it's, see here it is wrong. So, you see the ending here, it is sugar, and here also it is sugar, here also it is sugar, here very wrong representation. This should end in phosphate. When this ends in phosphate, this is correct, ends in sugar, whereas this will end in sugar, correct, and this will end in phosphate, which means now you add where it's phosphate it's 5 prime and where it's sugar it is 3 prime. Here it is 3 prime and here it is 5 prime. So, it is matching with the statement the 2 polynucleotide chains are running anti-parallelly. You can say it in brief. One is running in the 5 prime 3 prime direction, whereas the other is running the 3 prime 5 prime direction. That is called anti-parallel arrangement or alignment of the 2 polynucleotide chain. Next, third point is the point related to bonds, all the bonds. Now, I'll zoom this picture, focus on these components. Now, the bonds are going to be indicated in blue. The bond between the bases are the hydrogen bonds. See, these are the hydrogen bonds. So, what is indicated in blue is the hydrogen bonds, and there are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. The bond between the base and the sugar, so these, the base and the sugar, is called N-glycosidic linkage. Next, the bond between sugar and phosphate, see, that bond is known as Phosphor-Ester linkage. And the bond between that adjacent sugar, that is the phosphate with the adjacent sugar, again it is phosphor-ester linkage, but because it is making a second bond with another sugar, it's called phosphor diester linkage. So, according to the double helix model, the third postulate deals with bonds. Here you have to make a mention of 4 bonds. That is, the bond between the bases is the hydrogen bonds. And remember, between A and T it's double, and between G and C it's triple. Okay, that's one bond. Then the bond between the base and the sugar is N-glycosidic linkage. And the bond between the sugar and the phosphate is the phosphor ester linkage. And the phosphate with the neighboring sugar or with the adjacent sugar of the next nucleotide, it is called phosphor diester linkage. So, remember the bonds. If you have the structure, you can easily elucidate the bonds. So, the bonds between the bases, hydrogen bond. The bond between the base and the sugar, N-glycosidic linkage. The bond between the sugar and the phosphate, it is phosphor ester linkage. And that phosphate with the neighboring sugar, again phosphor ester linkage, but here it is called phosphor diester linkage. So, the third point is related to the bonds. Then the fourth point. The fourth point is connected to the turn or the coil. See, it is turned. You can turn, it is not ladder like, it is spirally coiled. It can be coiled either in the right-handed fashion or in the left-handed. In DNA it is coiled towards the right. So, it is coiled in the right-handed fashion towards the right. And if you analyze the coil, see with this picture, let's analyze this coil. See, it is coiled towards the right side or in the right-handed manner, okay, in the right-handed fashion. And one turn of the coil, see, one turn or one pitch of the coil, that length is 3.4 nanometers. Okay, one pitch or one turn is 3.4 nanometers. Once again, I am showing it here. One turn or one pitch. One turn or one pitch is 3.4 nanometers. And that one turn comprises how many base pairs? 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 10 base pairs. So, number of base pairs which are represented as BPs is 10. So, once again, our fourth point deals with the turn or the pitch of the DNA. The DNA is coiled towards the right, and each turn, the length of each turn is 3.4 nanometers, and each turn comprises roughly 10 bases. So, we can do a small calculation. So, one turn or one pitch is equal to 3.4 nanometers, and the number of base pairs in one turn is roughly 10 base pairs. Therefore, the distance between consecutive base pairs will be equal to 3.4 nanometers by 10, which is equal to 0.34 nanometers. This is very relevant because next topic we are going to calculate the length of the DNA. That time you should know the distance between consecutive base pairs of the DNA is 0.34 nanometers. How did we get it as 0.34 nanometers there? One turn, the length of one turn is 3.4 nanometers. The number of base pairs in each turn is 10 base pairs. So, therefore, the distance between base pairs is equal to 3.4 divided by 10, which is equal to 0.34 nanometers. And lastly, the fifth point, as told by Watson and Crick, is the stability of the DNA. DNA is very, very stable. When compared to RNA, DNA is very stable, and this, according to Watson and Crick, they said the stability is due to the stacked nature of the planes, the pairs. See, A, T, G, C, A, T, G, C. Look at the way they are stacked one above the other. So, the stacked nature confers stacked nature of the plane of the pairs of base pairs confers the stability. So, why they are stable? According to Watson and Crick, DNA is very stable because of the stacked nature of the planes of the bonds, hydrogen bonds. See, look at the stacked nature, one above the other, like a stack of chair. It's not easy to make to drop a stack of chair. They are so stable. No, there's a heavy breeze or wind or storm, a stack of chair remains very stable without falling. Whereas if it's single, it falls off fast. This is a complete stack. Okay, so this stacked nature of the planes of the hydrogen bonds confers stability to the DNA. This is the reason why DNA is very stable than RNA. There are other reasons also why DNA is more stable than RNA. When I deal with the topic DNA versus RNA, we can see the other factors which are contributing to the stability of the DNA. But as of now, from the salient features, or as according to Watson and Crick, stability is conferred due to the stacked nature of the hydrogen bonds. Clear? So, once again, brushing through the 5 postulates or salient features as according to Watson and Crick. 1. DNA comprises 2 polynucleotide chains and the sugar phosphate units form the backbone with the base protruding inwards. That is the first point. Second point, the 2 polynucleotide chains are running anti-parallelly, meaning one is running in the 5 prime 3 prime direction while the other is running in the 3 prime 5 prime direction. It can be represented like this. Okay. Third is regarding the bonds. The bond between the bases is the hydrogen bond. The bond between the base and the sugar is the N-glycosidic linkage. The bond between the sugar and the phosphate group is the phosphor ester linkage, and the bond between the phosphate group and the adjacent sugar is the phosphor diester linkage. So, the third point is related to the bonds. Fourth point is the turn. Which way is the DNA? It's like a ladder. As of now, I do like a straight ladder. It should be turned. It should be spirally coiled. It can be turned to the left or to the right. But DNA, it is coiled towards the right-handed side, or it is coiled in a right-handed manner, and each turn, one turn, the length of one turn is 3.4 nanometers, and roughly there are 10 base pairs in one turn. Therefore, the distance between consecutive base pairs in the DNA is equal to 3.4 divided by 10, which is equal to 0.34 nanometers. Very, very important to calculate the length of the DNA, these figures. And lastly, according to Watson and Crick, the stability of the DNA is due to the stacked nature of the bonds between the bases, the hydrogen bonds. That stacked nature gives the stability to the DNA. I hope the structure of the DNA as proposed by Watson and Crick is clear. Remember these 5 salient features. It should be written as it is because it was proposed by Watson and Crick in these 5 postulates. Next, we shall calculate the length of the DNA. Now, we are going to calculate the length of the DNA. The formula to calculate the length of the DNA is the total length of the DNA is equal to the total number of base pairs into the distance between base pairs. So, once again, the formula to find out the length of the DNA is the total length of the DNA is equal to the total number of base pairs in that cell into the distance between the base pairs. So, we are going to substitute. We should know this by heart. The total number of base pairs in human beings or in the cell of man is 6.6 into 10 raised to 9, which means 6.6 billion base pairs are there in the DNA. And that into the distance between consecutive base pairs we have just calculated earlier. What is the distance? It is 3.4 divided by 10, it is 0.34 nanometers. 0.34 nanometers. Now, this nanometers we cannot continue with nanometers, we have to give the SI unit, we have to go for meters. So, that is converted into meters. To convert it nanometers into meters, 10 raised to minus 9. We have to multiply it with minus 9. So, what happens? This get cancelled, this get cancelled, and 6.6 into 0.34, 6.6 into 0.34 which is approximately 2.2 meters. Just imagine what's the length of the DNA? 2.2 meters, as long as your mother's shawl. Okay, a shawl minimum has 2 meters. This is 2.2 meters long. So, very long DNA. Clear? So, once again, how we will calculate the length of the DNA? Total number of bases into distance between the consecutive base pairs. Substitute the total number of base pairs in the human DNA in the human cell is 6.6 into 10 raised to 9, and that into the distance between the base pairs, that is 0.34, and that when you convert the nanometer into meter, 10 raised to minus 9. So, this will get cancelled, this also will get cancelled. Finally, we have 6.6 into 0.34, we get 2.2 meters. So, the length of the DNA is very, very long as 2.2 meters. Now, it is mandatory to learn these figures also. You should know the number of bases, the number of base pairs. Base pairs can be indicated as BPS. In the bacteriophage 5 into 174, this is the name of a bacteriophage. The number of base pairs is 5386. Remember this number. Then the bacteriophage lambda 48502. E. coli 4.6 into 10 raised to 6, and man, as I told you, 6.6 into 10 raised to 9. Clear? This should be learned by heart. Okay. So, you can be able to calculate the length of the DNA. Just substitute this total number of base pairs into the distance between the base pairs. Distance between the base pairs also you should know. What's the distance between the base pairs? 0.34 nanometers. When you convert that 0.34 into meters, 0.34 into 10 raised to minus 9, then it becomes meters. Clear? So, this is warranted that you need to study these base pairs, these figures by heart. It is given in the text. So, once again, this is a bacteriophage called 5 into 174. The number of base pairs 5386. Then this is another bacteriophage called lambda 48502. Then E. coli, this is a bacterium, the base pairs 4.6 into 10 raised to 6, and man, the number of base pairs 6.6 into 10 raised to 9. This is very, very important. You should learn it by heart. Now, we understood and we have realized the structure, the length of the DNA is very, very lengthy when compared to the microscopic structure of the cell, and within the cell inside the nucleus, this lengthy DNA, which is as long as 2 meters, should be accommodated. See, 2 meters long, very long DNA should be accommodated inside this small dimension of the nucleus. Then how it is packaged? So, next topic we are going to see is the packaging of the DNA. Packaging of the DNA, as I told you, it is nearly 2.2 meter long in man. In prokaryotes like E. coli, it is 1.36 meters. Okay. Now, we are going to see how is this lengthy DNA accommodated inside the cell. Now, we know in prokaryotes there is no nucleus. Instead, there is a region called nucleoid. So, then how is this lengthy DNA accommodated? So, here you can see the picture of the prokaryotic cell with the chromosome, and look at the chromosomes. The chromosomes are arranged, the chromosomal DNA are arranged in the form of large loops. See, large loops. And how are these loops arranged as loops? Because they are connected by positively charged proteins. See, let these be the positively charged proteins. These positively charged proteins holds the DNA in the form of large loops. See, the positively charged proteins in this. The chromosomal DNA is arranged in the form of large loops held by positively charged proteins. Why should these proteins be positively charged? Because DNA is negatively charged. So, DNA which is negatively charged is held in position in the region called the nucleoid with the help of positively charged proteins. If these proteins were not there, what would have happened? They would have unentangled and they would have been lengthy and they would have been distributed and spread across the entire cytoplasm of the cell. But here we can see it is constricted only to the nucleus because of these positively charged proteins. It's just like pinning the ribbon to the notice board with the help of bell pins. The bell pins, you are making a flower or making loops with the bell pins. So like that they are pinned in the form of loops. I hope you have understood. In prokaryotic cell, there is no nucleus. Here the DNA is not spread across the cytoplasm. Instead, it is in the region called the nucleoid. It is arranged in the form of loops. And how it is arranged in the form of loops? Because it is held by positively charged proteins. DNA being negatively charged, they are held by positively charged proteins in the form of loops, large loops. So the answer to this packaging of DNA in prokaryotic cell is they are packaged in the form of large loops held by positively charged proteins. Clear? If these proteins were not there, if we remove these proteins, see I am removing these proteins, what would have happened to the DNA? They would have stretched and spread in the entire cytoplasm. But these are held in position with the help of these positively charged proteins. And remember, DNA is negatively charged. Now coming to eukaryotic, it is more complex. In eukaryotic DNA, the packaging is more complex. So I am starting from the simplest block and how it is being packaged. We know the size, we have analyzed and calculated the size of the DNA 2.2 meters. And how is this 2.2 meters accommodated in the nucleus? There are some positively charged proteins called histones. I am giving the sign positive inside this. They are globular proteins called histones. So this is one histone. These are positively charged proteins, and these proteins are positively charged because of the residue of certain amino acids like arginine and lysine. So we are going to learn nearly 20 amino acids in this chapter. As of now, I have introduced 2 amino acids, arginine and lysine. These arginine and lysine, because of the presence of arginine and lysine, the histones become positively charged. Now how many histone did I draw? One histone. It's like a globular protein. Okay. Like that way, the histones are getting organized into 8 such histones get organized together to form a histone octamer. So I am going to draw 8 such histones. 1, 2, 3, 4, 5, 6, 7, 8. A 3D structure. So I cannot show all 8 together. These histones, see, 8 of them are organized, and this structure is known as histone octamer. Now this histone octamer is positively charged because of arginine and lysine. Now the negatively charged DNA, suppose this white is the DNA, this negatively charged DNA wraps around this positively charged histone octamer, see, wraps around. Now the structure is called nucleosome. See, once again, the negatively charged DNA, see, this is the DNA, it wraps around the histone octamer, and now the structure is known as nucleosome. Now it is called nucleosome. So what is nucleosome? The DNA wrapped around the histone octamer is called a nucleosome. So like this really we can see the chromatin is arranged in the interphase stage. The DNA is found like this. Clear? So I am just drawing a number of such nucleosomes repeated. Now I just drew the DNA. Inside the DNA we have the histone octamer. See, 1, 2, 3, 4, 5, 6, 7, 8. Like that. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, you can see Histone Optimum. Histone Optimum. So you see how many nucleosomes, so we can identify how many nucleosomes in this rough diagram. 1 nucleosome, second nucleosome, third nucleosome, fourth nucleosome, fifth, sixth, 7, 8, 9. That way many nucleosomes are found in such a way that when you see under the electron microscope the interface stage, you will see the chromatin in the form of thread on string structure. It is known as bead on string structure. So this is the structure you observe under the electron microscope. And this phase is called the interface stage. See, it's just like the beads on string. This is referred to as beads on string structure. Clear? I hope you have understood how the DNA is packaged. Now, still this is which phase? This is representing which phase? Interphase. Now, just recollect the cell cycle. We have first the interphase stage, then we move to cell division where it is where it's mitosis, prophase, then from prophase we proceed to metaphase. Okay, at least in metaphase let us see the structure. So, in the interphase we see the chromatin is arranged like this. Okay. But as we proceed from one phase to the other phase, we are moving to the prophase stage. What happens to the chromatin threads? We have learned it gets condensed. When it gets condensed, it becomes shorter. In the prophase, what happens is that the chromatin gets condensed and becomes rod shaped. So, and becomes rod shaped. So, actually, suppose there were 4 chromosomes. Actually 4 chromosomes, you can see 4 rods. Again in metaphase what is happening? Further condensation is occurring. See, further condensation. To form that X-shaped structure, further condensation. Okay, to form the X-shaped structure. So, once again we can see the fine threads, elaborate threads and tangled mass called chromatin threads in the interphase stage, okay, where there is no packaging. But as we proceed to prophase, what happens to the DNA? It gets more and more condensed and coiled to form the prophase rod shaped chromosomes. Then again further in the metaphase further gets packaged, where the chromatin gets further packaged and appears as X-shaped chromosomes. Okay, this is the diagram which we have learnt in 11th standard. But as we move to the metaphase, this becomes thicker and shorter and becomes visible as X-shaped structures, the metacentric chromosome. Okay. If I revert to this, it is longer and less thicker, but whereas this one you can see the chromatin threads. I must make it very thin. 1, 2, 3 and 4. So, this is representing interphase, this is representing prophase and this is representing metaphase. Now, how is this packaging taking place? The chromatin threads further gets packed and packed. This is done with the help of another set of proteins called non-histone chromosome proteins. It is given by the abbreviation NHC proteins. So, these proteins are responsible for packaging of the chromatin to rods like structures and further packaging into X-shaped structures in the metaphase stage. Clear? So, I hope you have understood how the DNA is being packaged in the chromatin and how the chromatin is being packaged in the prophase stage to form the rod shaped chromosome, and further packaging and condensation in the metaphase stage. Clear? So, once again in a nutshell, explaining the packaging of the DNA in eukaryotes. So, once again I told you the DNA is very long as 2.2 meters long, then how it is accommodated in the nucleus. So, first you must talk about the histone, positively charged protein called histone. So, I am zooming the structure, a recap of what we dealt with. So, this is the histone. It is positively charged because of the presence of arginine and lysine, residues of arginine and lysine. Now, these histones get organized into histone optimer. What is histone optimer? 8 histones together forms a bigger protein called the histone optimer. So, 1, 2, 3, 4, 5, 6, 7, 8. This is called a histone optimer, and around this histone optimer the DNA wraps, and this structure is known as nucleosome. So, that way you can see the DNA goes around the number of histone optima, and you can see under the microscope, that too the electron microscope, the interface stage, it appears bead on string structure. Okay, I have showed you the photograph, the real photograph of bead on string structure. Okay, we see the chromatin at interface stage as bead on string structure. This is referred to as a famous bead on string structure. Okay, now this, you know, this is the first part of the cell cycle. First part of the cell cycle, which is called the interface stage. Now, next stage is these chromatin has to get packaged. Now, we are talking DNA packaging, we finished. Now, these chromatins should further get packaged. They should get packaged to form the rod-shaped chromosomes of prophase. Again, they should get further packaged to the X-shaped chromosomes of the metaphase stage. Okay, how is this packaging done? This packaging, further packaging is done with the help of proteins called non-histone chromosome proteins. Okay, so you can see these fine threads, to make the rod shape, they get packed like this way. Okay, and the non-histone proteins I will be putting like this way. They are getting packed like this. That way we get the rod shaped chromosome. See, now this is the rod shaped chromosome as we see in prophase. Now, as we move to metaphase, further condensation takes place. It's further packaging. So, it becomes more and more packaged and takes the shape of X. Okay, this is also done with the help of NHCs. Non-histone chromosome proteins are holding them and packaging them. That is why we get the X-shaped chromosome. Clear? So, the packaging of the chromatin to the chromosomes, the rod-shaped chromosome of prophase and finally the X-shaped chromosome of the metaphase stage is done with the help of proteins called non-histone chromosome proteins. Okay. Now, a small clipping to get an idea of the packaging. Okay. So, you can see 46 chromosomes distributed in the nucleus. See, we are zooming into the X-shaped chromosomes of the nucleus. Now, you can see each X-shaped chromosomes of the metaphase stage, it comprises the chromatin. See, the chromatin, see the way they are packaged? They are packaged, and the chromatin in turn is being packaged, see, DNA around the nucleosome. And look at the nucleosome, 8 histones together form a histone octamer, and the DNA wraps around the histone octamer. Clear? Now, coming to the same photograph of the chromatin. See, I told you the chromatin under the electron microscope is found in the interface stage. Now, observe this chromatin. I told you it is not just like fine network as we always refer network of thread. They are having beads, bead-like structure which are representing the nucleosomes. So, away from that, now I stain this. I am going to stain this. Now, you can see, does this part take equally the stain? No, because there are some regions which are densely coiled. See this region, concentrate at this region. This region is densely coiled, whereas this region is loosely coiled. So, I can identify in this chromatin itself, I can identify 2 types of chromatin. See, this is the chromatin. Generally this is the chromatin distribution, but here you can identify 2 regions. Here it is densely coiled. or densely packed, and this one, see, loosely packed. So when we add stain, this will take up the stain very dark. It will become very dark when you put the stain, whereas this one, the stain it takes up will be very light. Then another difference is that this particular chromatin, this part of the chromatin which is densely packed is called heterochromatin, and this chromatin which is loosely packed is called euchromatin. So now in this chromatin network, we identified two types of chromatins. Now the name I am going to give. This is heterochromatin and this is euchromatin. Okay. So the chromatin network which is densely coiled, see, it's densely coiled and therefore it takes up the stain very darkly, then it is known as heterochromatin. Whereas the other chromatin which is scattered, not densely coiled or not or loosely packed, it takes up the stain very lightly, and this is called euchromatin. Now we saw the two types of chromatin, heterochromatin and euchromatin. When we are going to deal with the next topic, we can find that heterochromatin is transcriptionally inactive, whereas euchromatin is transcriptionally active. So once again, if you want to find out the difference between chromatin, there are two types of chromatin. Within the chromatin network, you can identify two chromatins, heterochromatin and euchromatin. Heterochromatin, what is the features? They are tightly packed. Second, they take up the stain very dark and they are transcriptionally inactive. Whereas euchromatin is loosely packed. So when we stain, they take up the stain very lightly and they are transcriptionally active. So once again, in this chromatin bead on string, forget the bead on string, just take the entire chromatin network. In this network, you can see some regions are, if you identify the regions, you can see some regions are tightly packed, very tightly packed. See, very tightly packed is this region, whereas this is very loosely packed. This region is called heterochromatin, and this region is called euchromatin. And the difference between heterochromatin and euchromatin, one, the packing, this is densely packed, whereas this one loosely packed. Talking about the staining, this takes up the stain very darkly, whereas this takes it light. When we stain, it's very light. Then this heterochromatin is transcriptionally inactive, and this euchromatin is transcriptionally active. So we elicited the difference between heterochromatin and euchromatin within this photograph, which shows the chromatin at the interphase stage, also depicting bead on string structure. Okay, so that's all about the packaging of DNA. I hope you have understood the packaging of DNA in both the prokaryotes and eukaryotes. In prokaryotes, they just arrange the form of loops, large loops which are held by positively charged proteins. And in eukaryotes, remember, there are the histones, positively charged proteins, then a number of 8 of them organized together to form the histone optimum. Then on the histone optimum, the DNA wraps to form the nucleosome. Like that way, there are many nucleosomes, such a way that when you watch or see it under the microscope, the chromatin appears not like fine threads, it's like fine threads but beads on these threads, giving a bead on string appearance. Now this chromatin is found in the interphase stage. Now from the chromatin stage or this interphase stage, it has to proceed to the prophase stage, where you can see these chromatin thread condenses to take up rod-like structures. Then from prophase, it further condenses to become the X-shaped chromosomes of metaphase. How is the packaging there done? There it's done with a different set of proteins. The name of the protein is non-histone chromosome proteins, and they only bring out the packaging in the cell cycle from the chromatin stage to the rod-shaped stage of prophase to the X-shaped stage of the metaphase. Okay. So I hope this portion is clear to you. Thank you.